is Armaghan Sarvar, and I'm a Master of Bioinformatics student at the University of British Columbia and the Birol lab at UC Cancer Genome Sciences Center. Today I will talk about genome misassembly detection using STACH, a data structure based on stochastic type. The decrease in the cost of sequencing over the last decade has led to a more accessible generation of reads from DNA and RNA data. Many famous bioinformatics algorithms such as genome assembly or structural variation detection have been based on identifying short, fixed-length substrings of reads which are called k-mers. Hence, there has been a growing need for developing methods to store these k-mers that can support various queries. For example, as shown in this figure, the genome has been segmented into 3 mers, which will be stored to represent the genome. On the other hand, k-mer-based representations are not immune to sequencing errors, and that's why many tools and data structures have been based on using spade seeds, which can tolerate mismatches. For example, the Coombe filter is a probabilistic data structure used for fast membership queries that represents data by hashing values as bit array indices. An extension of the Coombe filter is the multi-net Coombe filter, or MIDF, which consists of a vector of identifiers together with the membership bits, and it stores spade seed sequences, an example of which has been shown here. Inspired by these data structures, we introduced a stochastic tile hashing or stashing in order to describe large collections of nucleotide sequences through a lossy representation. The proposed underlying data structure is called stash, which uses a fixed allocated memory and provides a means to store and query sequence mapping information without any alignment. For example, stash can answer the question of whether 2 sequences originate from similar genomic regions. But how is this data structure populated? The underlying structure of stash is a 2-dimensional bit array consisting of sets of T-bit tiles, where the rows or the memory loci are indicated by hashed values of subsequences. Having an input sequence, it is hashed using the anti-hash algorithm with sliding windows of symmetric spade seed patterns, and the resulting anti-hash values specify which rows of the memory block to update. Having h spade seed patterns, each sliding window position will update h rows of stash, and we call these h-accessed rows a stash frame. As for the next step in filling the stash tile, the column and the stored tile value are indicated by hashes of the sequence ID. So for the input sequence, the sequence ID is hashed twice using 2 different hash functions, and the resulting values are segmented into T1 and T2 bit tiles respectively. The tiles of the first hash function output specify the column, and the tiles of the second hash function output specify the value to be stored. In this slide, the whole data structure and the process of sequence data population have been shown for 4 spade seed patterns. In order to combine the anti-hash output values with the 2 sequence ID hashes, we can see that for the ith spade seed pattern, the ith most significant bits of the other spade seed pattern outputs are concatenated in order to address an index in the sequence ID hash type. So for example, here for the first hash function output, the first significant bits of the other hash function outputs are concatenated, which results in 0, 0, 1. So we will address index number 1 here, which leads to filling column 3 with the value 7. In order to answer the question of whether 2 genomic regions are covered by the same set of reads, we define a score metric called the number of matches that captures the similarity between any 2 frames of interest. And it is calculated such that each tile in the first frame is compared to h tiles in the second frame residing in the same column. And it is incremented if there is at least 1 equal value to the examining tile in the corresponding column of frame 2. In order to have a more general view of a genomic region and also ignore sequence errors and gaps, we define windows of frames, where each window consists of a set of frames that have a distance of stride with each other. We compare 2 windows by counting the maximum number of matches between all pairs of their frames. An expected range of values values for the number of matches is that for small distances on a genomic region, we expect the number of matches to be higher because the underlying frames would be coming from the same reads when filling stash. And for large distances, we expect this metric to be lower because there would be no connection between the underlying frames. As for the experiment, we have filled the stash with the packed bio hi-fi reads of the following human cell line. And by considering these reads, we choose a random position on them that helps us extract two windows that have a distance of delta with each other. By giving these windows as input to stash, we can calculate the number of matches between them. And by repeating this whole process several times, we get to a distribution of the number of matches. In this plot, we can see how by increasing delta values, which was the distance between two windows, the number of matches distributions get closer to the one of unrelated windows that have a distance of infinity. The red lines here show the confidence interval of 80% of the unrelated window matches, and this whole experiment shows us how stash is able to separate between related and unrelated windows. We can use this property of stash to deploy it in a wide variety of applications in genomics research. An example is genome misassembly detection and correction. As we know, content misassemblies can happen in assembly solutions due to reasons such as genome repeats and complexities. And the resulting misassemblies not only complicate downstream analysis, but also limit the contiguity of the assembly. As shown in this figure, by breaking misassemblies, we can correctly join the resulted subsequences during the scaffolding step. In order to perform genome misassembly detection using stash, after filling the data structure with a set of sequencing reads, by querying it with the corresponding assembly, we can extract the number of matches signal using two sliding windows of frames. Since we expect content misassemblies to have a low number of matches, we detect and cut such positions using our stash cut module. Here, we show the performance of stash in the task of genome misassembly detection when it has been filled with the same hi-fi reads as the previous experiment. Stash cut has been applied on the long-read SHISO assembly before and after scaffolding using the NT-link tool. In this plot, we show the number of misassemblies versus the NJ50 contiguity metric for the resulted assemblies. And these values have been reported by the cross tool. So as you can see, stash cut is able to outperform Tigment-Long, which is a state-of-the-art long-read misassembly detection method after the scaffolding step. And more specifically, the strategic cutting of stash cut has resulted in a more contiguous scaffolded assembly with a higher quality. Here we show the time and memory comparison results for stash and Tigment-Long. As we can see, stash uses a similar amount of memory, and although the complete pipeline of stash is currently slower than Tigment-Long, it still executes in less than 2 hours. To conclude, we have introduced a novel hash-based data structure called stash that provides an efficient representation of sequence data with demonstrated usage in identifying regional patterns and overlapping sequence frames without any need for alignment. We have also illustrated the utility of stash in detecting and correcting genome misassemblies. I would like to thank my colleagues, Lauren, René, and my supervisor, Dr. Inanc, Birol, and all the other Bioinformatics Technology Lab members who contributed to this project. My project funding is from NSERC, Genome Canada, Genome British Columbia, and the National Institutes of Health. Our software is available in this GitHub link, and you can contact me through this email address if you have any questions. Here you can also find the information about other posters from our lab. Thank you very much for your attention.